Great. I think we'll get started as people still come in. Hi, everyone, and thank you for joining our joint webinar with Rapid Nova and Illumix on new antibody characterization essentials from discovery to clinic. I am Genya, and I will be your moderator. We ask that if you have any questions during the webinar, that you use the Q&A chat and drop your questions there. And then towards the end of the presentation, we will hold a Q&A session. Our speakers today are Kyle, who is the Scientific Sales Representative of Canada with Rapid Nova, and we also have Hugo, who is the Chief Scientific Officer at Illumix. Kyle has been with Rapid Nova for 2 years and is dedicated to fostering collaborative partnerships that drive innovation and accelerate scientific progress in the field of antibody sequencing and characterization. Hugo is the Chief Scientific Officer at Illumix. Previously, he served as the CEO and co-founder of PhenoSwitch Bioscience, which was acquired by Illumix in 2022. During his PhD, Hugo used mass spec through different collaborations and core facility services to answer biological questions, where he learned that access to mass spec was far from simple. So those are your speakers for today. I will let them take it away with the presentation. And again, if you have any questions, feel free to drop them in the question box.
So I will present Alpaca first and then let Kyle do his presentation. So Alpaca is a Canadian-based CRO. We are a multi-omic CRO. We've developed and specialized into high-quality LC-MS solutions from sample prep to data insights. And the key to our success into this procedure is collaboration with our clients. We believe that's the only way that we can address complex scientific questions is by working hand to hand with them. So I'll let Kyle go on that.
Awesome. Thank you, Hugo. Okay, so hello, everyone. Thank you very much for joining us today. My name is Kyle. I am a scientific sales representative here at Rapid Nova, and I will be walking through the first half of the webinar on antibody characterization from discovery to clinic. To sort of start things out, I wanted to do a very brief intro as to who Rapid Nova is. So Rapid Nova was founded in 2015, and our technology is based on over 20 years of protein sequencing innovation. We have sequenced over 8,000 proteins to date and are steadily climbing up to 9,000 as we speak. At this current time, we are 75 plus employees, and we are the first to sequence a polyclonal antibody using only proteomics. I wanted to provide this quick timeline to give just a quick overview of what the process from discovery of a novel therapeutic to final patent filing and submission would look like, and where Rapid Nova and Illumix fit into this roadmap. So, of course, the timeline can differ depending on where in the preclinical pipeline your company falls and the resources that are made available. And I also wanted to highlight that I will be discussing the first 3 points listed here, then passing it over to our friends at Illumix for the remainder of the presentation. So when looking for a novel biologic, it will generally start with an immunization campaign against a target antigen. Once completed, our PAB sequencing service would allow for the identification of the most abundant mAbs in the immune repertoire. From there, you can study the kinetic activity of your top candidates through SPR. By studying the binding affinity and avidity, you can identify which candidates are best suited for targeted treatment or diagnostic applications. Once you've narrowed down your selection based on ideal binding parameters, epitope binding and mapping will allow you to study the binding location and conformational changes to ensure your lead candidates are stable and binding the appropriate epitope. So Rapid Nova specializes in proteomics and proprietary software for de novo sequencing of proteins and antibodies. Our REMAB monoclonal antibody sequencing is our foundational service and what we are really well known for. While the focus of today's presentation will be on our discovery workflow, I wanted to give an overview of our mAb sequencing, as knowing this is critical to understanding our polyclonal sequencing service. So essentially at the very start, what we do is we take a purified sample. We will always run a gel to ensure the sample meets our minimum sample purity requirements. Once confirmed, we perform a multi-enzyme digest, with each enzyme being specially chosen to have diversified cutting roles, providing peptide overlap. Samples are then loaded onto the mass spec. Spectra are generated and reassembled through our remab algorithm. Once a sequence is complete, we have a minimum of 2 checks through our bioinformatics team to ensure the primary sequence looks accurate. With over 8,500 sequences completed, our monoclonal sequencing knowledge and expertise has allowed us to expand into other services such as polyclonal sequencing, epitope mapping, and SPR, which I will be talking about today. So today I will be discussing a recent Alpaca case study that we had completed. This involved us isolating recombinant mAbs or nanobodies from an Alpaca PAB and studying them further with our characterization technology. So as noted, we are the first to study the sequence, or the first to sequence the Alpaca IgG proteome entirely de novo. And the following case study will walk you through our PAB sequencing protocol, including mAb isolation and screening, kinetic profiling of the top candidates, comparison of binding affinities through isoaffinity analysis, and epitope binning to study antibody interactions. Before we walk through the case study, I thought this graphic displayed a nice visual of the preclinical discovery process leading to clinic. So today, I will be discussing the second and third areas involving antibody discovery and expression, as well as lead characterization and selection. The case study will walk through a few of the many steps involved in preclinical drug discovery and validation. And I also want to briefly mention that this is, of course, a sample workflow. Not all workflows are built the same, but we look to utilize our expertise to help guide clients through this process. So step one within the case study was the sequencing of the polyclonal antibody. So we collected serum from an alpaca immunized with a rabbit polyclonal antibody. We then affinity purified it, fractionated, and digested the PAB. The sequencing of the alpaca PAB was performed using liquid chromatography tandem mass spectrometry, otherwise known as LC-MS. Now, this process follows the same as our mAb sequencing. However, there are many more mass spec runs and in-depth analysis being done on these samples. So it involves our sequencing platform and machine learning-based bioinformatic analysis. From there, we were able to compile the full amino acid sequences of alpaca IgG clones, including the chain pairing. We do, however, also have an optional proteogenomics branch as seen on the bottom of the slide here. So if available, we can take the blood PBMCs and do a bulk NGS on them. I wanted to highlight that the proteomics is still king here and still prominent. However, having some of the genetic data may just give us a better idea of sequences we were unsure about or ranked lower based on abundance and coverage. And referencing back to our Alpaca case study, this case study was done strictly using proteomics for the polyclonal sequencing. So results from the polyclonal sequencing. So after the PAB sequencing took place, there were a total of 10 sequences identified. 6 IgGs were selected for synthesis and further development. And the next step was to screen the mAbs for stability as well as affinity. So referencing back to our pipeline, we have gone through antibody discovery and expression and narrowed down our candidate mAbs. Next would be to characterize these based on the binding kinetics and location to the target antigen. So in our process, we now look to use SPR to measure both the K-on, K-off overall binding affinity to the target antigen of interest. So moving along, we now performed a screening and validation step. So we performed a yes-no binding screen through SPR with an ELISA affinity screen. The next step was to use SPR to measure the association rate K-on, dissociation rate K-off, and affinity KD. So the 2 rabbit mAb targets were P17 and CD3 epsilon, and they were chosen based on the antigen pool from the alpaca immunization. And there were a range of affinities reported to the 2 targets. So we'll quickly review our SPR workflow and highlight some of the reasons why we use our instrument for antibodies. So at this time, we utilize the Nicoya Open SPR with gold nanoparticles to measure antibody antigen binding kinetics. So what this allows for is it enables us with the possibility of immobilizing binding partners in both orientations. So whether that be your antigen of interest or antibody being immobilized, with the opposite being run over as an analyte on the sensor chip. And the chip is being selected based on parameters of each ligand itself. The system is highly sensitive, generating an optimal signal to noise ratio, and is calibrated by our on-site experts for accurate data analysis and insight. We can work with antibodies, antibody fragments, peptide antigens, and large complex protein antigens, even in protein complexes. So with that in mind, in this case, we immobilize both P17 and CD3 epsilon, as seen here under the ligand side, onto our SPR instrument and ran each of the 6 MABs to measure the binding kinetics. So out of the 6, 4 were selected for further profiling based on their binding affinity. Those most notably were R1, R4, R7, and R9, as you can see in the top graph, or the P17 graph. R4 and R9 actually displayed binding to both of the target antigens, while R1 and R7 showed selectivity just to P17. So next, what we wanted to do was to compare the binding affinity of each clone through an isoaffinity analysis to convey the data. So we performed an isoaffinity analysis to dissect individual contributions of each of the clones. So each of the respective charts shows the binding affinity with respect to the 2 different antigens, P17 on the left side and CD3 epsilon on the right. So the binding affinity, also known as the KD, is the dissociation rate, or the x-axis in both charts, divided by the association rate, or the y-axis in both charts. And an affinity differing by 1 full magnitude can be seen by each of the diagonal lines on the charts. So some notable observations included R4 and R9 having a very similar binding affinity to P17, but differing by a full magnitude in CD3 epsilon, even with the same association rate. So as you can see here, in reference to P17, R4 and R9 both bound with a very similar on rate as well as off rate, R4 is being slightly quicker. However, in CD3 epsilon, they actually differed by 1 full magnitude depicted here. So very similar on rate, but R9 had a significantly quicker off rate. Another notable observation is R1 also had a 20 times lower affinity despite a comparable on rate to R4 and R9. So as you can see here, lines up quite well. And this is actually just based on its pretty fast dissociation rate or off rate. So next, what we looked at, we looked at the clone's epitopic diversity through epitope binning. So for epitope binning, there are multiple different ways to go about the process. This does ultimately depend on the biochemical properties of the antigen as well as the end goal of development. So some antigens can prove to be more difficult to work with, resulting in off-target binding if run as an analyte or as a ligand. Others will sometimes undergo conformational changes based on the orientation in which they are used in the kinetic analysis. So in this study, we evaluated how antibodies can bind to respective epitopes in pairs through a tandem binning approach, as seen involving the assay setup here. So each clone was injected at a saturating concentration of 100 nanomolar concentration, which was found as the saturation concentration required to block the epitope for each of the antibodies. So from there, each clone was used as either a saturating antibody, displayed here, or as a binding antibody or an analyte, displayed here in a 4x4 binning matrix to show changes in binding. So any inhibitory function would result in incompatible clones and place them in the same bin. So some notable observations that we found were that R7 was a unidirectional clone, meaning it was a strong binder as an analyte, as depicted here, but would actually block other clones when used as the initial saturating antibody. So very good binder when used as an analyte, but again, would block R1, R4, and R9 respectively. So this would result in asymmetric blocking. So R4 also displayed some interesting data. Based on the epitope's location, it would allow for clones R1, R7, and R9 to each bind, but was almost completely blocked out when run as an analyte, with each respective clone first being used as a saturating antibody. So again, would allow for R1, R7, and R9 to bind in a tandem approach, but when run as an analyte was unable to bind with R1, R7, and R9 already present. So this data paints a partial picture. However, to fully understand what is happening with the epitope and paratope, we needed to perform epitope mapping. So we performed epitope mapping through HDX-MS, and when coupled with epitope binning, this data allows for accurate understanding of the binding location. It not only shows the area that an antibody is binding to an antigen, but any conformational changes that may take place along with binding. So our platform actually allows us to work with a few different special cases. These include disordered proteins, membrane proteins, as well as multi-protein complexes. And oftentimes this data is very important in IND filings as well as IP claims to show how your molecule is different from those with existing patents. Here is a very rough overview of the process. So essentially what we do is we're able to measure exposure levels of hydrogen atoms on the surface of the protein and protein complex. So any exposed hydrogen readily exchanges with deuterium, while hydrogen atoms buried in the protein structure are not readily exchanged and can hint to us where the antigen binds. Examining the peptides through bottom-up mass spec and with a provided amino acid sequence allows us to determine which regions are binding locations. So I wanted to quickly show the 3D model of the P17 antigen. As you can see, the 4 clones are actually mapped out, showing the binding location to the respective antigens. So you can see R1 here in this purple color, R4 at the bottom here in orange, R7 and R9 respectively. And this was all done in-house by our HDX-MS epitope mapping team. So finally, just to go over a few different applications of epitope mapping, of course, antibody-antigen epitope mapping is a popular choice. It also is applicable to biosimilar characterization and comparability, batch-to-batch reproducibility for antibody production, antibody stability analysis, and antibody engineering and optimization. So finally, I wanted to quickly re-summarize the funnel that we had previously been discussing or walking through. So here at Rapid Novor, we look to help researchers with their early preclinical research on biologics. This includes our antibody discovery services. So in this case, we isolated recombinant mAbs from an alpaca PAB that was immunized with polyclonal rabbit antigens. We then selected the best clones, characterized them by SPR to determine the overall binding affinity. We plotted the clones on isoaffinity plots to visualize any similarities or differences. And then we took this data and performed binning on the top clones. This showed us any competing clones that would not be compatible through tandem binning based on the orientation of the binding ligand and analyte. So I would now like to pass the presentation over to our friends at Illumix, who will be able to provide downstream data on top candidates, continuing down the preclinical funnel closer in the next steps towards IND submission. So I will pass it over to Hugo.
Give me 2 seconds. The prism. Sorry. Sorry about that. Somehow presenter mode started with the presenter mode full screen and not the actual presentation. Anyway, so I'll start right away from there. So like Kyle presented, so RapidNova can do a lot of things right here when you are selecting your target, building your antibody, and selecting what would be your lead candidate antibody biologics for development. Once you've got this and you know where it binds, then next step is to produce it. You want to do preclinical studies, you want to inject animals, and ultimately go to the clinic. So what we can help you with at Alpaca is working into that characterization during the process of production. So optimization of production, optimization of the formulation, optimization of the sequence for stability, and also looking at the low-level contaminants, HCP, PTMs, amino acid variants that can be introduced during that process. And after that, you will go to the clinic, which would be GMP lot release, which is a totally different thing that we won't discuss today. Okay, so the idea of biologics characterization, people know LCMS protein sequencing, but let's think about, well, let's go beyond just the primary sequence confirmation. You already know your primary sequence. You've done that with RapidNova. You understand what will be the sequence, but then when you produce it, there's a bunch of things that can happen. Among them, important characteristic that will be looked upon will be oxidation. So here I present an example of primary sequence, and then you will have an oxidation with a certain rate of oxidation. Then you will have deamination, and then you need to confirm that deamination is actually there, because you rely on type 2 spectrum match software that will tell you things at a certain level of confidence, and then you need to confirm the data. You will also look at your protein structure, making sure that the cysteine bridge that you have within your biologic are always the same, and the amount of free cysteine and bound cysteine are there. So dipeptide characterization, cysteine bridge characterization. You will look at the glycan, where they are, what composition they have. And that's pretty important, because when you submit to the FDA, they want to make sure that your molecule is always the same and always the same unique molecule that you're giving to your patients. And you will look at some degradation and in C-terminal clipping. So that's the kind of thing that you will be looking at. And how you do it. So people know bottom up, but you also do intact mass, where you just inject the whole antibody itself, or the reduced form, or reduced in deglycosylated form. And why you do both is actually because one will inform the other and vice versa. So that's a good way to actually validate your data. And we can help you with that. And here I will present you how we can help you and why it's important to, it's not that easy sometimes. So I'll give you the example of deaminated peptide. So here you've got that amino acid, asparagine can be deaminated and will actually convert by a loss of water here and a delta of 1 Dalton into aspartic acid or isoaspartic acid. So a delta of 1 Dalton. And this is very important regarding stability of the antibody. But think of it, this is a pH-dependent reaction. So you also have to have a sample prep workflow that will control very tightly your pH. Otherwise, you will see that happening just during the reaction of your sample prep. And once you got this, so that's a typical peptide spectra that you get there. You've got the precursor, and then you also have the isotopic distribution being distributed there. And what you will note is that there's actually M+1, which is exactly the same Dalton, not exactly the same, but nearly the same Dalton difference that you have here. So typically you will see the parent, and if you trust the software the software will tell you, hey, actually you've got the deaminated peptide here, But that's an error, because if you look at the distribution, it's exactly the same isotopic distribution that you got there, and also exactly the same retention time. So you need to look into the software and validate it, and you will realize that deamination is actually that small peak right here. So that's the kind of thing that can be, you can help the software to make the right assessment there, especially using retention time prediction. But typically, software will do that kind of errors, and you can report false claims there, and it's important to review the data correctly. The next topic I wanted to cover was, it's a hot topic because it's highly discussed, and the FDA just released a new recommendation regarding LC-MS for whole cell protein. So they just approved one drug that was submitted to them using LC-MS only. So typically people use ELISA to look at whole cell proteins, but ELISAs have their limitation. And they will also look at sequence variant, where you will have ribosomal leaking or primary sequence mutation that will create sequence variants, single amino acid variants. So here, in that case, the FDA will recommend to report anything that is above 1 to 100 nanogram per milligram, which is 0.1%. And 0.1%, if you look at this is 3 log. It's quite a big difference. But still, this 0.1% can be dangerous. And if you think of this, when you do your production and when you do your validation, you want actually to look a little bit below that, because those are contaminants or variants of concerns. So you actually want to go below 0.01 and actually identify them and then track them in time. And the challenge here is the high level of a major protein is pretty much like plasma proteomics. So you've got one major antibody that is there, very high abundance, and you're only searching on that small search space of that antibody. And you tell the software, hey, by the way, look into this and try to find any contaminants that are low-level contaminants. And what we see is that typically the software uses a false discovery rate to report its confidence into peptide to spectral match, PSM. The issue is the small search space is not appropriate to that approach. The other thing that we see often is, for example, self-assignated contaminant. Because of that high abundance of a spectra, you will see some peptides being assigned, but actually it's actually the antibody that's being assigned. I will show you a bit why. So here, that's a typical MS-MS spectra for peptide. That's a predicted spectra using machine learning. And you will have a B ion series and a Y ion series, which is actually how it fragments from one end or the other end of the peptide. And this is the composed MS-MS spectrum here. And you see the dynamic range. So we go to 250K of intensities, and some, you see the protein there, you don't see it because it's just so low there. So if we zoom in, that's a 3 log zoom in, okay? And we still see good information there. What will happen with PSM, the software, because of the search space, and if you don't tune it properly. So we've got the sequence here, and what it does is measure the distance between the MS-MS spectrum to assign the peptide. So we've got the DE of peptide here, we've got IDE of peptide here. But actually, because the software doesn't have anything to correct itself, the search space is so small, it will start to do some creative assignation of amino acids and create some false assumptions. And here, I'm only speaking about Y and B ions, which is the major ones. But once again, remember, we are in the 3 log zoom in, and there are other ions that are less common but still present that can create that kind of confusion with the software. So that's very important to adjust and adapt properly. And here I will present you a case where it was published wrongly because of that kind of assumptions. So this is a paper that has been published on the NIST monoclonal antibody by Burris et al. They roughly use 2 labs to sequence the NIST reference monoclonal antibody to look for single amino acid variants. And what we see there is that only from the 2 labs, only 14 of this list was detected in both labs. Only 1 is confirmed by IRES instrument. Only 9 are confirmed by 2 sets of orthogonal enzymatic digestion. And if we look at the data, they didn't have whole cell proteins or contaminants into their FASTA when they did the screen. So what we did is we repeated the same experiment, but we added actually 3 other enzymes to make sure that we just back every claim we have with at least 1 enzyme. So we've got trypsin, we've got gluci, we've got chymotrypsin. So we've got different overlapping peptides, pretty much like what Rapid Nova does when they do de novo sequencing. They want several confirmation of the information. What ended up is that, and we added the solid proteins and the contaminant in the search. So first thing is that we do identify 2 host cell proteins that are quite abundant on 5 peptides. Those peptides could have been assigned actually to those variants there. So that's the first big difference. If we look at this, and we have developed a very strict criteria for selection and cleaning to remove any false positive, none of those actually pass. And even worse than that, if we take those sequence and we add it to the original search, we don't tell the software, look for any modifications. We tell them, look for those. The software is not even able to see those because we changed the parameters there. But we did identify 4 other single amino acid variants, but they were below 0.02%. So they would not have been reported to the FDA ultimately. So that's very important there. And if we look back at the data again, and we look at how we screen and how we filter, and we look at the mass that they were reporting, so we see that those mass actually represent most of them artifacts. So incomplete calculation, CH2 loss in the source, formaldehyde loss in the source, C2H4 loss in the source. We've got also a formulation that is a common artifact that we see in mass spec, and we also see other alkylation products. So the key here is that, well, when you do that, you need to have your search engine to work properly. You need to filter for the false positive. You need orthogonal validation, and you need IRES instrument, because in the original paper, they were using old linear ion trap, which is actually not IRES and can actually make even worse false assumptions there. And I've got time a little bit, so I will present you also cool stuff that we can do. It's not related to single variant analysis, not related to HCP, but it's one of the advantages of working with biology, is that here we present a data from proteomics, standard proteomics. And what a client asked us was, could we do the PK and the exposure at the same time? And the answer is yes. So what we did is we spiked in some internal standards and samples, and we did the acquisition standard proteomic, very nice profile, treated, untreated, vehicle control. And then we did automate the standard curve without any method development there. We just use internal standard peptides and just report that. And we still have a pretty neat quantification in the samples from 25 nanogram per ml to 500 nanogram per ml. No method development at all, but you still have, I think it's very cool when you do some early analysis and you want to do exposure and get data. That's one thing that you cannot do with small molecule. You can do it with proteomic experiment. That's the kind of thing that we've done. Well, thank you. I think it will be time for questions.
Awesome. Thank you to our speakers for a great presentation. Here's your contact info. And then again, if you have any questions, feel free to use the Q&A box to ask those questions. We do have a couple in there right now. So the first question, I believe this one would be for Kyle, is are you doing anti-ID antibodies since the antigen is the antibody?
Yeah, good question. I don't have the specific details surrounding the rabbit PAB antigen that we did use for the immunization campaign, but that would certainly be something to look into further to verify that.
And then the following question of that is, of those 2 different antigens in that pool for the Alpaca immunization, which one was used in the binning and epitope mapping?
Yeah, so apologies if I didn't explain that in the presentation, but it was the P17 because we did find a response from all 4 clones to P17. CD3 epsilon was only the 2 when we had mapped them. So it was to the P17 antibody or antigen.
Any other questions to come in? Question for Hugo. Do you regularly use orthogonal digests in your work?
Well, for PTM analysis and sequence coverage, yes. That said, sometimes trypsin alone is good enough, or modified trypsin workflow is good enough, depending on what you want to do. I would say if you want to go in very confident data, very confident assignation, at first, I would say ordinal digest, and then once you have confirmed it and you just want to track it, then you can do just one of the digestion set to track and follow.
Any other questions coming in? Yes, the webinar will be made as a recording that should be sent out to you shortly after we finish up the live webinar. But yes, it will be a recording.
Feel free to write to us both with questions after.
Okay. Doesn't seem like we have too many questions coming in. We can, I guess, stay around for a little bit longer. Again, contact infos are there. So you're more than welcome to send Hugo or Kyle your questions afterwards, especially if you're watching the recording. Another question is for Hugo, how long does your analysis take? A week, more or less? So what is the timeline?
It really depends. Yeah, well, the timeline depends on what you ask. So if it's just PTM analysis, the workflow with orthogonal digest, the timeline is suiting. So we generally say it's a 6-week from sample reception to delivery date that we give, but we can go faster than that, and it doesn't take 6 weeks. And the key and the difficulty is mostly in the reporting and the threshold that will be set. So what do you want do you want to see very low level of PTMs, or okay, we have the 0.1%. 0.1% is easier to report and takes less time. For the single amino variant and the HCP, typically requires a little bit more manual validation because it's really background information, and I will not be comfortable just reporting what the software tells. You need to do some manual. It takes a little bit longer.
Okay, it seems like that is it. So thank you for everyone for joining us today. Again, this will be made as a recording, and please take note of the contact info if you do have any questions later on. Otherwise, thank you for joining us today.
Thank you.
Thank you, everyone. Take care.
You too.